All right, welcome back, everyone. Today we're going to be doing a lesson on transcription. So, what is transcription? Let's go over here. Oops, excuse me. So, here I have the central dogma, if you look at the box, of biology, basically. Um, we know that DNA encodes genes, those genes, and that deoxyribonucleic acid composed of nucleotides is transcribed into RNA, ribonucleic acid. So it gets the hydroxyl group put onto that ribose sugar. Then that RNA is translated into protein. And that protein enacts its function amongst the cell, anywhere within and sometimes outside. Okay, so transcription. Transcription is the process of turning DNA into mRNA, which involves basically copying the DNA's instructions and making it into a form that can be exported out of the nucleus. You don't want your DNA going out of the nucleus because it would be degraded by the cell and lysosomes and other cell, components that are designed to remove these extraneous or foreign substances from the cytoplasm. Therefore, we send copies of DNA, known as RNA, into the cytoplasm to reach these ribosomes or the ER, if that's where it's going. So, how do we regulate this process of transcription? We have three major ways of doing this. We have enhancers. These are cis-acting DNA sequences. They can either be near the coding DNA or they can be far away from the coding DNA. I think the limit is 1,000,000 base pairs, but I'm not sure. I know that these enhancers are DNA sequences that can increase the transcription of genes. So if you need to upregulate a gene, you need more protein, you're going to upregulate that gene. That gene might be the responsibility of an enhancer. And that an important note is that these enhancers do not have to be directly near these promoters to enact their effects. We also have silencers or repressors. I'll just add that there. So silencers slash repressors are regulatory DNA elements that reduce transcription from their target promoters. They can do this by inhibiting access to the promoter or just blocking the promoter. Or just silencing that gene, preventing transcription. Lastly, but not least, we have the promoter region. This is the region required to catalyze transcription. So it's a nucleotide sequence, a series of nucleotides in the DNA where the RNA polymerase binds to catalyze transcription. You see here, it marked. Oops. Let's go down. Silencers and repressors can obstruct the promoter region, preventing RNA polymerase from binding to the promoter sequence and begin transcription. Additionally, initiation catalyzes. So there's three phases of this process of transcribing DNA. We have initiation. So let's just focus right here. So we have initiation. This catalyzes DNA transcription and RNA synthesis. Promoters can be a 35 nucleotide region, a 10 nucleotide region, or a +1 region. And this is the transcription start site. So those promoters we were talking about earlier, which catalyze genetic transcription, so these promoters, they can be a 35 nucleotide region, a 10 nucleotide region, or a +1 region in prokaryotes. And prokaryotes are cells that don't have a nucleus before the nucleus. And they utilize an RNA polymerase holoenzyme that consists of subunits 2 alpha, beta, beta prime, and sigma, if you want more detail. In eukaryotes, we have sequences of nucleotides that signify the start of the promoter region. So we have the ta-ta box, which is thymine, adenine, thymine, adenine. So it's thymine, adenine, repeat. And they call them box. Yeah, that's just what it is. Then you have the cytosine, adenine, adenine, thymine box. Then you have the guanine, cytosine box. And these are all identification areas where RNA polymerase II, which transcribes mRNA, we'll go over the different RNA polymerases shortly, which transcribes the DNA. So this can happen by something like a transcription factor binding to the promoter region. But the TATA box, the CAT box, and the GC box all give like signals to show that this is where you start. Yeah, they're identification areas. Okay. And here we have repressors. Again, they can bind to the promoter region and prevent the transcription factors from binding and start signaling the start of transcription, or RNA polymerase from binding, which also starts transcription. And transcription factors generally move from outside the nucleus into the nucleus to bind to the DNA. They're activators that help bind RNA polymerase to the promoter region. So, hey, I need more of this protein. Boom. That respective transcription factor moves from the cytoplasm into the nucleus, binds to the DNA, signals the start of transcription. Now you're on your way to make more of that protein. And that is the extent of initiation. So, moving on. Now, I know this is going to be a lot, but we're going to get through it with ease. So, I mentioned earlier that there were several types of RNA polymerase. What is RNA polymerase? RNA polymerase takes DNA, and polymerase is an enzyme that reads DNA. I think that's more accurate. But they do different things based on the type. So RNA polymerase type 1 synthesize, it's part of the ribosome. So RNA polymerase 1 and ribosomal RNA are the primary components of ribosomes. So in the ribosome, you'll have RNA polymerase 1 working with rRNA to catalyze protein synthesis. If you want to know how that works, just go to the translation video, or there's probably more information on YouTube if you want to look up just RNA polymerase 1 or ribosomal RNA, or ribosome function for that matter, or another translation video. But that's translation. So RNA polymerase 1, translation, rRNA. So think of it just like that. RNA polymerase 1, R, ribosome. Easy. So the one we're going to focus on mainly today is RNA polymerase 2. This is for messenger RNA. mRNA, messenger RNA, conveys genetic information from DNA to the ribosome. So like I said, you need to make a copy of the DNA because you don't want your DNA getting into the cytoplasm. So that DNA is read by RNA polymerase 2, which then synthesizes mRNA and sends that mRNA after it gets post-transcriptionally modified into the cytoplasm towards a ribosome or the endoplasmic reticulum. Okay. Lastly, but not least, RNA polymerase 3. RNA polymerase 3 works mainly with tRNA, and that's transfer RNA. And we know that the tRNA from the translation video basically matches the codon to the anticodon and this respective amino acid in protein synthesis. So here it says transfer RNA is an adapter molecule composed of RNA that carries amino acids to the ribosomes as directed by codons in mRNA. And then this is going to be important later, but I would just take a note of this, but I will go over it later. RNA polymerases 2 and 3 have small nuclear RNA, and this is used in eukaryotic splicing. So that'll be important later. So now that we know the enzyme that's responsible for actually transcribing this DNA, what is the process? So the process is called elongation. This word right here, elongation. You're elongating the mRNA strand as you copy the DNA off the DNA template. Um, so part of this, let me actually write this because I want to give you a visual. I feel like a visual would be very useful here. Um, so we're gonna have our. Oh, that's horrible. Okay, we're gonna have our. Where is the crayon? There it is. Okay. So we're going to have our DNA strand here. We're going to call this the coding strand. We're going to have our template or antisense strand down here. So let's label that. It's easy to follow along. Coding strand. Coding strand. And down here, let's make that smaller. Coding strand. Easy. Down here, we have our antisense or template. So this antisense or template strand is going to be this one. And for the sake of, for the sake of this, we're going to have this one going 5 prime, this one 3 prime. This one's 3 prime. And this is going to be, 5 prime. So I don't know if you've watched a translation video, so I'm going to assume you haven't. And just state that the 5 prime is going to have the phosphate group. That's going to be important later. Because that's what's at the 5 prime end. If you've looked at our DNA video, which would be very useful to watch before you try to watch transcription, because it all works together. It builds upon itself, which is why I can't go over scientific papers unless we get context to the scientific papers by starting from square one. And this is square one. So up here 4 minus, our phosphate groups in there. Boom. Merry Christmas. Okay. So we have our, we're going to add our nucleotides. So we got a nucleotide. Boom, 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 boom. And let's say this is a C. Let's see, this is G. C. Let's do this one A. Let's do, get a T in there. And then this one could do whatever. Let's make it a A. So this one, that's a C. This is going to be a G. Oh my God, that's horrible. Okay. We're going to do this way. Boom. There it is. G. I apologize. This was another one. Boom. God, I'm drawn. So that's an A. So we're going to put a T there. Adenine, thymine, thymine, adenine. Like once you get it, you kind of just oops shit. Excuse me. Probably gonna have to edit that out. Okay. Oh my goodness. Why did it just? Okay. Yeah, I'm gonna have to find a new way to do these videos because this is kind of bad. Okay. 8. Boom. And then, last but not least, another thymine. So this is our DNA. We have our coding strand. We have our on the top. We have our antisense or template strand on the bottom. Now, mRNA is going to be synthesized in the 5 to 3 direction because polymerase. So we're going to go up here to the elongation. Back to this. RNA polymerase 2, which synthesizes mRNA, functions by opening up this DNA. So it's going to sever all the hydrogen bonds here so that it can start reading from 3 to 5 and synthesizing our mRNA. So it unwinds and breaks the hydrogen bonds, like helicase in DNA replication, which will be another video. It reads the DNA from the 3' to the 5'. So it starts with the hydroxyl side and moves to the phosphate side. And that's basically because as you add DNA to that side, it's going to target in the coding strand, it targets the hydroxyl group with a phosphate group. So 3 with the hydroxyl, it adds a phosphate, which adds a substitute nucleotide and has an open exposed hydroxyl group again. And then just adds another phosphate group. And so you're aiming to basically phosphorylate those hydroxyl groups, but not actually. It's not. That's not how it works. But you can think of it like that. Actually, you probably don't want to think about it like that because why not? You're creating the phosphate backbone. Boom. There you go. Okay. So then RNA polymerase is going to be this guy right here. It's going to be this little enzyme. After it's separated the DNA strand and it's got this open single strand, it's going to stabilize that single strand of mRNA because it's synthesized. As it moves in this direction, oh goodness. As it moves in the 3 to 5 direction, it's going to synthesize an mRNA that is synthesized 5 to 3. Because if this is the 3 end, we know that it's going to read it and split out the complementary base pair. So it's going to synthesize an mRNA. It's going to read that. It's going to pop out a C. It's going to see another G. It's going to pop out a cytosine. It's going to see a thymine. It's going to pop out an adenine. Now, in DNA, we know that the complementary base pair is a thymine for adenine. But in mRNA, we know that it's going to make a uracil. So that's a major differentiating factor between mRNA and DNA. So then you're going to have thymine there, and it's going to make an A. So now the RNA polymerase has moved down this line and it's synthesized this mRNA strand here on the bottom, right here, this. And when it gets to the end, it's going to see a termination sequence, but we'll get into that later. Right now, we're just doing elongation. So that's kind of the general mechanism. I just wanted to like draw a picture so that it makes a little bit more sense than me just reading these lines. So as we know, RNA polymerase 2 opens the DNA, it stabilizes the single strand of mRNA, it unwinds and breaks the hydrogen bonds like helicase and DNA replication. Excuse me. It reads the DNA from the 3 to 5 on the template strand and synthesizes the mRNA from 5' to 3', which is from phosphate to hydroxyl group. It adds phosphates to the hydroxyl groups. And lastly, it's not agreed upon on whether it is capable of nucleotide correction at polymerization site 1 nucleotide back. So I did some reading and I saw this and I was just like, it looks like it's not agreed upon, but there's people doing tests to see whether RNA polymerase 2 is capable of proofreading and correcting its, mRNA strand or DNA strand. But it's not decided upon yet. So that's all for elongation. Once you get that, it's kind of straightforward. It reads it from 3 to 5, it synthesizes it 5 to 3, and it's it creates this mRNA strand that we see down here. So once it gets to the end, it's called termination. Termination. So we have initiation, elongation, and termination. And there's two kinds of termination in prokaryotes. One is Rho dependent, which removes RNA polymerase from the DNA. So there's a protein called Rho, RHO, and it is moving. So let's label these again. We're going to have 5 prime on the coding strand. For the sake of continuity, we're going to make them all the same. 5 prime, 3 prime. I have 3 prime, 5 prime. Boom. Yeah. Okay. The top strand will be the coding strand, and the bottom one will be the template strand. Boom. Okay. So it's going down. RNA polymerase is doing its thing. It's transcribing. It's making those thymines into adenines, making those adenines into uracil, making those cytosines into guanines, and those guanines into cytosines. And eventually, once it hits the Rho is moving up the mRNA strand. So the strand that's being synthesized, Rho is just like tailing it almost. So it's like literally riding that strand. So Rho is like right here. So RNA polymerase is synthesizing on the template strand, which is the bottom strand here, and Rho is moving up that template strand. And then once it catches the polymerase right here, it's gonna boot it off. So it removes RNA polymerase from the DNA, like forces it off. It's like, hey, I caught you. It's time to move. Usually, I think that's because the gene has been transcribed or whatever coding sequence needed to be coded or transcribed has been transcribed, and Rho meets up with it and just moves it off. Then there's Rho independent termination. So it doesn't use Rho. Rho is no longer used. So we're just going to get rid of all this. Yeah. Boom. Don't need you. Okay. Rho independent termination. There are these guanine and cytosine rich regions in the mRNA. So in the DNA, it'll probably say CGGG, and then in the mRNA, it'll be like GGG, CCC, but it'll be repeating. And in between those, there'll be like a bunch of random nucleotides. So let's say this is the sequence. It'll be like, we'll have like A or something like that in between. And then because there's a gap, and we know that adenine forms 2 hydrogen bonds with thymine. So 2 hydrogen bonds, and guanine forms 3 with cytosine. So the guanine and cytosine bonds are stronger. And because there's so many of them, they're going to fold in and connect, kind of ignoring those adenine and thymine connections. And because of that, it forms like a loop that connects like this. So you'll have your A's up here, and then you'll have your G's and your C's over here. And once this loop forms, you have cleavage enzymes that just samurai slice that mRNA to signify the end of transcription. That's in prokaryotes. It's called the hairpin loop, and it results from GC repeats, guanine cytosine repeats. In eukaryotes, it is different. In eukaryotes, we have something called a polyadenylation signal. So if we go back to this previous drawing, I intentionally made these last three adenine and uracil because you both was thinking ahead. So I did that because a polyadenylation signal is when, instead of, you know how in prokaryotes you have the GC rich region, in eukaryotes it's a polyadenylation, so it's an adenine rich region, and that once the RNA or mRNA or the RNA polymerase starts reading this, it reads that and it's like, okay, we're done, and it signals cleavage enzymes to samurai slice that sequence, right? And then the mRNA is free to do what it wants. Go to post-transcriptional modification sequence. So that's basically termination in a nutshell. Just remember, in prokaryotes, you have the guanine and the cytosine-rich regions that form the hairpin loop. And then in the eukaryotes, you have that polyadenylation signal, which is going to be important here. I'll just go right into it then. At the 3' OH end, you have those polyadenosine tails from polyadenylation. Remember, those AAUUAAs, that adenine-rich location, and it's got a lot of uracil, and that signals an enzyme called poly-A polymerase, polyadenine polymerase. And that helps initiate the translation process once it reaches the ribosomes. It also helps prevent degradation of newly synthesized mRNA by nuclease enzymes, you know, those cleanup cells that are just wandering around the nucleus like, hey, you're not DNA, what are you doing here? You know, they be protecting that DNA, man. Like, the nucleus is no joke. So you gotta think, like, if I had the secret formula for a Krabby Patty in here, like, would I really be letting anybody just in here? No. So you have to market. You have to be like, hey, I work here, you know? And that's pretty much what the polyadenosine tails from the polyadenylation signal do. Next, on the other end, on the 5' end of the newly synthesized mRNA, you have heterogeneous nuclear RNA. Wait, no, excuse me, I misspoke. You have, so before these post-transcriptional modifications occur, it's called heterogeneous nuclear RNA, which means it's basically, it's different nuclear RNA. It's not, it's like immature. It's not mature. It's not ready to ship out to the ribosomes yet. That's not supposed to be there. Sorry, I was distracted. Okay, so on the other end, so we just talked about the 3' polyadenosine tails, which signal to prevent degradation and signal to initiate translation upon reaching the ribosome. You have the triphosphate group, which is added by RNA triphosphatase. Because RNA triphosphatase cleaves off one phosphate group from the triphosphate group that is added to the 5' end. Then, so as you know, because there's a 3 triphosphate group on the 5' end of the newly synthesized mRNA, and you cleave off one, now you have 2. So there's 2 phosphate groups on that 5' end. So 5' end, you got a phosphate group. There's 3 of them, triphosphate. Yep. And then you're gonna have another one. But you kind of get the point, right? So it's like there's 3. Ooh, this is not the right software for this. So you have 3 RNA triphosphatase is going to cleave off this one. It's going to get rid of it. And then guano. Sorry about that. Reached my 30-minute timer faster than I anticipated. That keeps happening. I got to talk faster. So we ended off with the triphosphate group on the 5' end of the newly synthesized heterogeneous nuclear RNA molecule. The RNA triphosphate cleaves off the third phosphate from the triphosphate group, and then guanylyl transferase transfers guanosine monophosphate. So guanosine molecule and one mono phosphate to that. Um, well, it transfers the guanosine monophosphate to that um, diphosphate group, making it three phosphates again. Um, Attached to a guanine molecule. But it does, this process takes energy and takes the breakdown of guanosine triphosphate into guanosine monophosphate and two phosphate groups. The two phosphate groups just, get discarded. The guanosine monophosphate gets added to the heterogeneous nuclear RNA on the 5' end. Finally, methyl transferase, it's an enzyme that transfers methyls, adds a methyl group, and the product is called 7-methylguanosine cap, and that's on the 5' end of the heterogeneous nuclear RNA. So now you have a 7-methylguanosine cap, which helps initiate the translation process, like the other post-transcriptional modification down here, and they both do the same thing, more or less. So they help initiate the translation process, and they help prevent degradation of newly synthesized mRNA, heterogeneous nuclear RNA, by nuclease enzymes. So in eukaryotic DNA, or mRNA, you have exons and you have introns. So exons code for amino acids. As you know, nucleotides are grouped into pairs of 3 called codons that signal for respective amino acids. Introns are non-coding DNA. They're in between the coding DNA, that's how I remember it, which do not code for amino acids. Now, remember earlier we said that RNA polymerase 2 and 3 were capable of synthesizing small nuclear RNA? We did set that up here. Bang. 2 and 3 are capable of synthesizing small nuclear RNA. I said make a note of it. This is why. Small nuclear ribonuclear proteins are responsible for the splicing. Because if you have a bunch of directions in your Krabby Patty secret formula, going back to that analogy for the DNA, you're not gonna want that. You Mr. Krabs out here. You're not trying to have nothing extra. Bare minimum, maximize profit. So you're gonna get rid of those introns. You're gonna excise those introns, and then you're gonna stitch together or splice the exons together. And if you look at this beautiful drawing over here, Pablo Picasso over here, you're going to see that the splice site begins at the end of the exon 3'. So we know that the 5' is the phosphate, and we know that the 3' is the hydroxyl group, and we're going to put that here for the exon 1. You see exon 1 here, got it labeled. That's what it is. Okay. And then on exon 2, we should just label this now, since we're doing it. 5', 4. I'm gonna keep on doing that because it's really important. That's like fundamental to a lot of genetic and DNA oriented activities. I don't know what grade my audience is going to be in, but I do know that that's important to know. 5' is phosphate, 3' is OH, and I also know that it took me it made genetics a lot harder since I didn't know that when I was going through it, but hindsight is 2020. So then you have this intron, And at the beginning of this intron, so right after you get through the coding DNA, you have this unit that marks spot of the splice site. It's a guanine and a uracil. So a guanine and a uracil molecule mark the beginning of the splice site. So on the exon side of that GU is the splice site. The GU marks the intron, and then at the end you have a G, which marks the beginning or the end of the intron, and on the other side is the beginning of the coding DNA exon. So how does this process actually happen? So the small nuclear ribonuclear proteins bind to the intron right here, and they just kind of chill there until it's time to go. And when it's time to go, they go right to the splice site, and they just cleave it. So now there's a break. So the exon has been separated from the intron on the 3' end of the exon, the coding DNA, and the 5' end of the intron. And what's going to happen here is there is a deliberate adenine molecule in the very center. It's a branching adenine. So if you can imagine that, that's what's going to happen. And the 5' end of that, uh, guanine is, it's looking for an OH molecule because now it's free, and it's gonna attack that deliberately placed branching hydroxyl group in the middle of the intron. It's gonna kind of loop into itself. Um, so if you go down, you'll see that right here. It's gonna look like this, right here, something like that. That's all facilitated by the small nuclear ribonuclear proteins. Um, but so now you got this half loop, and what you're going to need is the splice site to be, what's it called? Let's see. Uh, yeah. Oh, yeah. So then you have this 3' end. I don't know. That was silly. I didn't even look at that. So 3', the OH. Like eventually, this stuff just comes intuitively, but you know we got to get there first. So the OH or the 3' end then is now looking, so it doesn't have a phosphate group to bind to. Oh wait, yes it does. On the other side of that guanine molecule, there's a 5' end of the next exon. It's going to attack that and squeeze this loop out, and that is going to be excised essentially, or spliced. And then the OH stretches, reaches the next phosphate group, and now you have a complete coding strand, um, a complete mRNA coding strand, a mature mRNA that has been post-transcriptionally modified. Um, yeah, to have the complete amino exon. So finally, we have alternate splicing or alternative splicing. Let me see. I think it's alternative. Yeah, it's not alternate. Sorry about that. Alternative. Okay, so alternative splicing is basically a post-transcriptional modification that allows eukaryotes to have like nuance in their proteins. Excuse me. So post-transcriptional modification produces variants of a protein through splicing at different exon sites or splice sites, splicing sites, and connecting different exons together. So the coding strands, you know how in this example here, we had this exon and this intron, this exon. But what if there was like another intron or an exon over here? And what if there's an exon over here, you know? So there's an intron and then an exon. And I'm just going to put I for intron, intron, you know? Boom. And then put E for exon, coding strand. Boom. And what if we're like, oh, we don't need this protein. We need that protein. So, and that protein is different from this protein. So instead of, connecting here, it's gonna go around that exon, around that intron, and boom. I, E. Oh, that was rough. E. Oh. And stretch those two together so that now you have exon 1 and exon 3 connected together. And that's going to translate for a different protein because the codons are going to be different. So that's alternative splicing. And I don't have an example, but if you look it up, like there's many examples of alternative splicing in eukaryotes. And that's important to know, that eukaryotes are responsible for alternative splicing. Okay, next. Alternative splicing. Now we have, lastly but not least, we have RNA editing. RNA editing is another post-transcriptional modification of mRNA through a process such as like deamination, removing the amine group from the amino acid. If you're familiar with amino acids, you know that they have an amine group, a carboxyl group, an alpha carbon, and a side chain. And an RNA editing example would be like deaminating the nucleotide, or the deamination of the nucleotide. Yes. No. Yeah. Yeah. Deamination of the nucleotide, like the base. And essentially changing the identity of that nucleotide to change the resulting codon into a different codon. So an example of this is uh, cytidine, which is a nucleoside because it's not attached to a phosphate backbone. It is a nucleotide that is not in a DNA molecule, or so it's not embedded into the phosphate backbone. So it's a nucleoside. So cytidine deaminates the uh, the cytosine amino group and substitutes uridine for cytidine in hepatocytes. So that's one example. So hepato, hepa is the liver, and you're going to substitute a uridine, which is a uracil nucleoside, for cytidine. And you can see here, if this is the codon, right? If you have a CAA codon and you change it into a UAA codon, that's a stop codon. So that could truncate the translation sequence, or the mRNA sequence, and change the function of the protein, which is important and has a variety of uses. So overall, that's the process, a really nutshell process of transcription. But I hope you found this video useful, and I'll see you in the next one.